Okay, so today I'm going to talk about the work that we've been doing with this splicing factor PRP40A in the context of early spliceosomal assemblies. I'm going to give some examples of what different techniques within NMR and with other biophysical techniques are useful to understand how this protein interacts with another splicing factor. Okay, so a bit of biology. I hope you remember from your biology lectures. So splicing is one of the major processes that happens to the pre-messenger RNA in eukaryotes, together with of capping, the 5' capping and 3' polyadenylation. Splicing is one of the more dramatic changes that the pre-messenger RNA had, in which large portions of the sequence, the introns, are removed, and the leftovers, the exons, are linked together in order to give rise to the right coding sequence to produce the proteins. You can imagine this is heavily regulated, and in eukaryotes it's important because also it spans the gene variability. So from one single gene, we can have different versions, which are called isoforms. Quick example of fast pre-messenger RNA. We have different exons, and if exon 6, for example, is included, what we have at the end is a protein, membrane protein, which has a pro-apoptotic function. If the exon 6 is excluded, is skipped, then we have a soluble protein that remains in the cytoplasm, and then it has the opposite function, is anti-apoptotic. So alternative splicing can lead to very dramatic changes in the cells. Errors in the regulation, errors in splicing can lead to different diseases, and also it's been related with cancer progression and metastasis. Okay, how splicing works is quite complicated, and I'm not going to go through it, and it involves a lot of proteins and ribonucleoprotein particles, this U1 to 5, that are loaded into the RNA, into the intron for each splicing event in a sequential manner, very well organized. And at the end, what we get is the removal of the intron and the link of the two exons after two transesterification reactions. There are different checkpoints for regulation along the cycle, but we are interested in the group of analyzing what's happening at the beginning, because it's in this early stage of the splicing assembly where the different sequences that undergo splicing are recognized and are selected. Again, this is quite complicated. I'm not going into biology details, but there are many different factors that recognize the cis elements in the RNA sequence, in the boundaries, the splicing sites. They are constitutive, there are alternative splicing regulators that promote or inhibit the interaction, and also there is a special kind of splicing regulators that are bridge proteins that connect one splicing site with the adjacent through the exon, which is called exon definition and is quite important in higher eukaryotes, and also through the intron. And this is important as well because at the end in the splicing cycle, these two regions have to be close in order to do the reaction. So the protein of interest, the PRP40 of this talk, is one of these type of bridges. It's a cross-intron bridge protein that is essential in yeast, but in humans is considered more as an alternative factor, only affected a certain subset of splicing events, mainly those related to microexons, and these are a type of exon which are really short, and it's because the link between through the exon cannot happen because it's too small to assemble all the proteins in the short sequence. Okay, this protein contains two different regions, an N-terminal region containing two WW domains that interact with the C-terminal part of this protein, SF1, which recognizes the branch point sequence, and then a C-terminal region. Today for this talk, I'm going to focus only on this type of domain. The WW domain was quite popular in the late 90s and 2000s. Several aromatic residues that recognize peptides. But this recognition is quite promiscuous. It's not very selective normally. In the case of the WW domains for PRP40, the first one, it's known that has some preference for a PPLP motif. I forgot to mention that normally the peptides that are recognized are proline rich. Okay, and the second W domain has not preference for binding. But what is known in the literature is that using the tandem of PRP40, there was also a different binding. There was binding to different proteins that are not splicing related. So they are quite promiscuous. They're binding huntingtin, forming, and other proteins not related to the splicing process. So our main goal of this work is trying to understand how the WW tandem is arranged and how can selectively bind to SF1 if it does so. And moreover, we went beyond that and started to analyze the function of the whole N-terminal region, because we discovered some regulatory regions there. Okay, let's start from the beginning. And we did the canonical approach to the study of the tandem with the different constructs we analyzed by NMR. And we can see from the relay of these different constructs that the two WW domains are folded independently and they don't have interdomain contacts between them. When we analyze the chemical, the assignment of the tandem, we can see that apart from the canonical fold of each domain with the 3 beta strands, we have 2 secondary structural elements, 2 helixes that are part of the fold. We use normal NMR techniques with acquisition of different noses, and we solve the structure of the tandem, which has this elongated shape with the canonical fold for the first WW domain, and the second one separated by a linker region with a turn and helix. And the C-terminal helix is interacting with the linker one. We did not detect any interdomain NOS. Therefore, when we analyze the ensemble of the structure, we have this situation. The relative orientation of one domain respect to the other is not fixed. And this is mainly driven by the lack of data, okay? But this could reflect that there is some flexibility between the two domains. For that, we did more experiments. We went back to the thermonuclear NOE measurement, and we realized that the linker region has lower NOE values, reflecting that the linker could be more dynamic. We also acquired RDC data, which will give us an idea about the orientation of the different domains. So we measure the tandem in the alignment medium with phages, and we use just the data from the first and the second W domain, the signals coming from these two domains, and fit it to the structure of those domains. The fitting, as you can see, is really nice, but the tensor definition for each one is quite different. This could indicate that the orientation, the respective orientation of one domain to the other is not conserved. They have different various orientation. So indicating again that it could be flexible. Of course, we fit the data to the ensemble that we have. The fitting is really bad because we have several, random orientations of one domain respect to another. We did acquire also paramagnetic relaxation enhancement data, in which we label several positions in the tandem with a spin label. It has an ampere electron, and this produces relaxation in the signals that are in a certain range. This is using NMR to get long range distances. According to our ensemble, there should not be major interdomain contacts, but as you can see, when we label one domain, we do see relaxation in the other domain. So with this relaxation, we believe it's coming from the flexibility of the tandem. So there is a certain point in which the spin level is close to the other domain, and this is reflected in the relaxation data. We also acquired SAX data, as we've seen before. The SAX is a nice technique that gives us an idea about the shape and the size of the particles that we have in solution. From the distance distribution function derived from the SAX data, we can see that we have a bimodular function with one major peak coming or populated by the intra-domain distances, and shoulder that would reflect that there are longer interdomain distances. This is a typical situation when we have more elongated shape like the one that we have. When we do the fitting of the ensemble, we can see that the fitting is quite nice. And of course, we are not sampling the whole conformations that can be in solution, but we can give us an idea that this is what probably we have in solution. Okay, so we conclude that we have a very dynamic tandem, sampling different conformations, but when we check in literature, we saw that the GIST version is rigid. the two domains, the orientation of both domains is fixed. And this is mainly coming, what we think is coming from the linker region. When we check the sequence, the linker, the helix in the linker in humans is broken by a proline, which is located here. And this disconnects the N-terminal part of the helix, making this turn, with the rest. And probably this is the source of the flexibility that we observe. Okay, so analyzing first how the tandem looks like in solution, then we move to the analysis of the interaction with SF1. It's known that PRP40 interacts with SF1 through the C-terminal part, which is believed to be intrinsically disordered and containing quite a lot of prolines. WW domains are known to interact with polyprolines, and before starting to analyzing the interaction with SF1, we did a more thorough approach with artificial peptides. So we created link to the GB1 protein, C-terminally different lengths of polyprolines, from 6 prolines to 29 in a row. And then we use ITC to characterize the interaction with the tandem. As you can see here, we have different ITC titrations, and the analysis of all the different titrations show that with increasing number of prolines in the peptide, we had increasing affinity until we reach the number of 16 prolines in a row. And then after that, what we have is an apparent decrease in affinity, but an increase in the stoichiometry. So 16 is the minimal length of polyprolines that is able to interact with the tandem at the same time, with both domains at the same time. And if we more prolines, if we make it longer, what we have is the apparition of a second binding event in the suboptimal affinity. Okay, we also follow the interaction by NMR, and here I'm showing you the titration with a 6 proline peptide and with a 16. You can clearly see that the 6 proline peptide is showing titration in a fastest chain, while the 16 is in the intermediate close to slowest chain, indicating higher affinity. When we map the interaction in the tandem, we detect chemical sheet perturbation in the interface, the canonical interface, the beta sheet. But for the case of the 16 proline peptide, we also detect chemical sheet perturbation in the linker. We assign this to the following. So when we have a 6 proline peptide, we have one molecule binding to one WW domain and another molecule binding to the second. And probably the conformation of the tandem is not really affected. But when we have a longer peptide, we have simultaneous binding of the peptide to both domains at the same time. And this probably will constrain the conformations that the tandem can happen, and the linker region is affected. And that's why we see the perturbation here, and also in the C-terminal helix that interacts with the linker. We also discard that there is a direct binding of the linker to the polyprolines. Okay, then knowing that 16 MERS is the minimal length for binding to both WW domains, we went back to SF1 sequence, and we searched for different peptides here. We did the same approach with the GB1 and with ITC, and we started with the titration. We did titration with some peptides that contain space prolines that show no interaction or very low affinity. Then there were other peptides that contain 2 proline tracts broken at some point by the presence of another type of amino acid that show an affinity around 20-30 micromolar. When these 2 proline tracts are completed, what we have is an increase in affinity up to 10. But sampling the whole SF1 C-terminal region, we only found one peptide with significantly higher affinity, which is this one order of magnitude more, around 1 micromolar. So we believe that this is the anchor point of the PRP40 tandem in SF1. If we do titration with longer peptides containing this binding site motif, we have basically similar affinity. And if we do titrations with other type of peptides, we have lower affinity. In this case, the affinity resembles the one that we had with the 16 MERS that contain 2 polyproline tracts. If we start adding more part of the C-terminal, what we obtain, for example, in here is more or less the same affinity in the low micromolar range, but an increase in the stoichiometry. And if we keep increasing the length, we have the same situation until we do the titration with the whole C-terminal part of SF1. So we conclude that this is the binding site of PRP40 within SF1, but there is some promiscuity with 1 order of magnitude difference in affinity to other regions in the C-terminal part. We wanted to characterize how the binding is happening, and for that, we first check how the flexibility dynamics of this complex are. So we did the same approach as for the APO. We purify the complex, and we acquire the RDC data in the presence of phages. Again, we took just the data from the first and the second WW domains, and we fitted the tensor, and we obtained really nice fitting, although the structure of that we were using were the APO structures. So it means that the backbone of the WW domains are not affected upon binding. But now we have a different situation. Now the tensor definition for both domains is quite close, with a magnitude of around 11 Hz, and the rhombicity is quite close, meaning that now there is a correlation of both orientations. So what we conclude is that probably the two domains now are fixed. The orientation of both domains is fixed in solution. We also analyzed the heteronuclear data, and we saw that the linker region now gets more rigid, has higher values of heteronuclear NOI. And also we acquired SAX data, and now the complex in blue looks more compact and has less contribution of these two bimodal distribution. So we have a complex that is quite stable, the affinity is high, and it seems quite rigid. The way to go is trying to get the structure by crystallography. But as in the case of LoRa, we could not get nice crystals to get the structure. So then what we did to get the structure is NMR. And we were really lucky because the peptide did not contain any aromatic, and we did different filter noces. And you can see here in this example how we get signals from the peptide in this dimension and give a lot of NOE data, NOEs cross-peaks with the aromatic side chains from the peptide. We did different combination of labeling plus different NOEs, and we got the structure of the complex, also together with the RDC data. We have the first WW domain interacting with the N-terminal region that contains this PPLP motif, and the second domain interacting with just the C-terminal part of the peptide. At the beginning, we thought that the binding for both domains is the canonical, because it's as it follows. So the canonical binding of a WW domain. We have one proline here inserted in the hydrophobic core of the WW domain with these three aromatic residues. The second proline is sticking out, solvent exposed. Then the third position is different between both domains. We have a leucine and a proline, both inserted in another hydrophobic group. And then we have the last residue, this proline, which is stacking with together with the tryptophan. But for the case of the first WW domain, we observe more contacts. We have this sequence, proline, proline, glycine, alanine, proline, wrapping around the tryptophan and stacking the two tryptophans, the two prolines sandwiching the tryptophan in the ring. And also we have extra contacts from the backbone. This type of interaction has not been seen for the WW domains before, and we believe this is the cause of the increased affinity that this tandem shows, and also explains why is the selectivity for this type of sequence. Okay, we did also validation of the structure with the PRE data. And here now we can see that the domain, the complex is more rigid, and there is no inter-domain contacts, because now the PRE data for the boundary state shows no inter-domain relaxation. And also the SAX data that we acquired for the complex, now it fits really well to the complex, the structure that we saw. Okay, so this is what we did with both proteins, but then we wanted to see how the interaction is happening in the context of the whole N-terminal part of PRP40, because we found that N-terminal to the tandem, there is a region enriched in prolines. And when we created the construct of the first 300 residues, we saw that two things. First, that the main region that is folded is the tandem, but the signals from the tandem, here overlaid with just the tandem spectra, they are shifted. They are not in the original position. And when we narrow the construct, we see exactly the same, which resembles these shifts are similar to the ones that we obtained when we titrated the tandem with a peptide, with a protein-rich peptide. So what we believe is happening is that the N-terminal region is folding back and interacting with the tandem. We wanted first to understand which motifs in the N-terminal part are interacting. For that, we did a titration of this construct with the high affinity peptide from SF1. And then we monitor the chemical shift perturbation. Of course, we have quite a lot of perturbation in the tandem, but also we observe that signals close to these two motifs in the N-terminal region were shifted as well, from a folded chemical shift to a more random Coy values. We confirm that these two motifs are the ones that are interacting by mutating to alanine, and those shifts that we observed when we extended the tandem N-terminally are now coming back when we mutate the two motifs in the same construct. Okay, so we know that this is happening, but what's the effect on binding to the peptides? For that, we went back to ITC. And this is the data that we have from ITC of the tandem to the high affinity peptide and to another suboptimal peptide. There is a 1 order of magnitude difference. But when we do the same experiment in the presence of the N-terminal part, so with this construct, we have reduction on affinity in both cases. But in the case of the high affinity peptide, still we have a low micromolar KD. But in the case of the suboptimal peptide, we cannot really determine the KD, but we can estimate it in the hundreds of micromolar range. So very dramatic change. When we mutate these two regions, we again recapitulate the same binding affinity as with the tandem domain. So what we think is that this N-terminal region acts as a regulatory region that inhibits the interaction for suboptimal peptides, for basically other non-specific targets. We asked help to our collaborators in Mainz, and they did a CoIP using two versions of the N-terminal part of the protein, the wild type and the mutant, and they expressed them in HeLa cells, and did the copurification and analyzed which proteins are copurified with them. Simply, they observed that there was a huge enrichment of proteins in the case of the mutant, meaning that there was a lot of unspecific binding. There were some proteins that are not really related to PRP40, so there were quite a lot of unspecific binding. So with this, I finished the summary. We've studied the tandem of WW domains, PRP40, the structure and the dynamics. There was a previous structure that was shown, but it was truncated, prematurely truncated, and well, I didn't talk about that, but we confirmed that this is the right one, taking into account the dynamics. Then also we identified the SF1 binding region, and we explained why it has higher affinity than other protein-rich peptides, and solved the structure of the complex. And more important, we saw that there is a regulation, there is an N-terminal part that acts as an autoinhibitory peptide that modulates the selectivity towards the right targets. And importantly, we found that in humans, there is like around 40 proteins containing 2, 3, or 4 WW domains. And almost all of them contain adjacent N or C-terminal rich protein-rich regions. And we postulate that probably they are acting in the same way. So you have the selectivity of each domain, you increase the selectivity by putting them in tandem, and also you reduce unspecificity by adding these regulatory regions adjacent to it. So with this, I'm finishing, and I just want to thank the people involved in the project, Michael, for letting me the second postdoc in his group, and also all the students that helped me with the work, and our collaborators in Mainz as well. and all of you for your attention.